Okay, so now for today's lessons or these lessons, we're going to look at the translocation. So particularly this translocation take place in the phloem and is described as the transport of soluble organic substances within a plant. So basically what we want to learn here is we want the soluble organic substances, or in this case, we must be able to know that it must be water soluble, not lipid soluble. It must be water soluble. Okay, and these soluble organic substances are referred as assimilate. So in our syllabus, we are going to use sucrose, okay, as our model, but don't forget other assimilate as well. For example, amino acids, okay, those can be assimilates. So the assimilate are translocated the directions always from the source to the sink. So what is the source? When use the word source, basically means that it's the place where we supply or plant supply the assimilate, and we use the word sink to describe the place where this assimilate is received. Okay, and we need to know that sometimes the translocation take place. The translocation take place. This part is bidirectional. You know what's bidirectional? So means that it can actually go. Sorry, it can go up. Okay, it can go up the phloem's safety element. It can go down. So it depends on the hydrostatic pressure gradient. So let me use one example potato tubule to show you a potato tubule to show you, okay, what is meant by the source and what is meant by the sink. Different season, okay, particularly for potato tubule, at different season, you can see that the source and sink refer to the different parts of the plant. It's not like in xylem always unilateral or unidirectional, only from the root to the shoots. Okay, so let us look at the during summer. Sorry. During summer, you have the potato plant. Okay, so potato plant. Okay, with the leaf. Okay, then we have the soil and you have the underground stem. Okay, underground stem for potato. So if you look at the leaf carry out photosynthesis. So produce sucrose, for example, or assimilate. So means that, can I say, the leaf now carry out photosynthesis during summer because high light density. Okay, so therefore this part of the plant is termed as the source. The leaf act as a source. Okay, so this photosynthetic product assimilates now will translocated down to the root to store, okay, to underground stem. Are you clear? Go down to the stem, underground stem. So what's happened to this underground stem? Underground stem now store, receive and store the assimilate. Okay, so you can see that when you store assimilate, they receive, right? So means that this one is the sink. How I say, from the source to the sink. The source of the leaf, underground stem is the sink. So therefore, you can see that potato is formed. Potato tubule is formed because it stores the starch. You can see that these underground stems, okay, start to grow and store this potato tubule. Okay, so now after summer, it comes to the season like autumn of winter. Okay, skip the autumn, like winter. So where the farmers start to harvest the potato. Okay, so in the winter, so they harvest the potato, keep the potato, and then during the next year, spring. Okay, next year, spring. What will happen here is the farmers start to plant the potato tuber. Can I see that? So when plant in potato tuber, so the plant start to grow, right? So the plant still cannot carry out photosynthesis yet. So why? Because no leaf, correct or not? So it means that this potato tuber now have the storage of starch, correct or not? So it's supply. So the hydrolysis of starch. You see that give the plant assimilate. So this assimilate now move from the potato tuber, now go up to the stem. Can I say that direction different already? Just during the summer goes now, okay, and then during the spring the directions up. Okay, so in this case you can see that the potato tuber now act as the source and the stem receive, right? So the stem now act as the sink. Can you see that? So this is to show that the direction is bidirectional. Now, this bidirectional also occur within the same season. Now, what's mean within the same season? Now, you look at this. So this is the plant. A sieve tube element. So plants, every plant, they don't have only one sieve tube element. They won't have one sieve tube element. So this first sieve tube element. So if this is where the leaf is, then you have the flower. And we have the roots. Yeah, sorry, let me draw this. Okay, draw the roots. Okay, so what will happen here is you can see that the assimilate here, they act as the source. Here can be the sink. Flower cannot carry out photosynthesis, right? The roots can act as a sink. So within these plants, they have many sieve tube elements. So we have the sieve tube elements number 2, sieve tube element number 1. So depending on the pressure gradients, you can see that the assimilate can actually transfer from the leaf to the flower at the seed. So going up, okay? It might, okay, these assimilates might go down to the roots, but can you see that they use 2 different sieve tube elements? Are you clear? So it means that at particular time, if the transportation from the source to the seed is going up, then it will go up. So it won't have 2 directions at same sieve tube element. Are you clear? So it means plants at one particular vascular bundle, they have many sieve tube elements. So some in charge for transporting the assimilates up the plants, some may transfer down the plant. So this is bidirectional as contrast to the xylem. You look at the xylem, so it always from the root to the shoot. Mass flow. Are you clear? So in this case, this one called unidirectional. Okay, so stop recording. Okay, now we continue with this translocations mechanism. So similar to the xylem, again, is by mass flow, not active transport, not the diffusion because too slow. So it by mass flow. Okay, so if one look at this mass flow, we need to have the pressure gradients, or more precisely hydrostatic pressure gradient. And in this case, the hydrostatic pressure gradient is due to the osmosis. So it's osmotically generated pressure gradient. Between where? Between the source and the sink. Okay, in order to achieve this, then we have to use the active transport by using ATP to generate this pressure gradient. Okay, so this model is termed as the pressure flow model. Okay, so let us look at what actually happened. So let me draw this structure, sieve tube elements. So this is sieve tube element. So sieve tube elements at the top part here near to the source. And we have the sieve tube elements at the bottom here near to the sink. Okay, so both of them, I say that sieve tube element will have the component cells. So component cell CC, component cell. Okay, and then at the source here, you have the mesophyll cell. So this is mesophyll cell. Okay, and then at the sink, we have the potato tuber, for example, at the roots. Okay, so we have the cortical cells for storage. Okay, or cortex, cortical cells. Okay, so what actually happened first? Because these mesophyll cells carry out photosynthesis. Okay, cortical cells carry out storage. They act as a storage cell. So what happened? Photosynthesis produce assimilate. So our assimilate here is sucrose. Okay, assimilate sucrose. So this sucrose now enter into the sieve tube element. So first process. So look at the first process. First process is an active loading of assimilate, which is sucrose in our context here, into sieve tube element. Okay, near to the source. Okay, near to the source. So what actually happened? What is the consequence here? So because sucrose, they are water soluble, so reduce the water potential at the sieve tube elements near to the source. You see that? So now first thing, water potential become lower. And then we do know that at the vascular bundle, beside you have the xylem. So this is xylem vessel element. Xylem vessel element. So what will happen here, because water potential inside the sieve tube element is low, so water molecule now move in by osmosis. So process number 2, osmosis takes place. So it means that water molecule now enter into the sieve tube element at the source. Okay, so osmosis takes place down the water potential gradient. So what is the consequence here? So the consequence here, we increase the volume of the fluid. Therefore, we increase the hydrostatic pressure. Because due to what? Due to accumulation of water, now you can see that hydrostatic pressure is high. Okay, because water molecule enter. Okay, what happened at the sink? At the sink, so this sucrose now unloading. So number 3. What's number 3? Unloading of assimilate of sucrose. Okay, out of sieve tube elements at the sink. Okay, so you remove solute. So means that water potential now is high. Compare here, water potential is low. Okay, so therefore water molecule now move out. So this number 4, osmosis again. So due to the loss of water decreases the hydrostatic pressure. So it means that now hydrostatic pressure. Let's see that. So in this case, it generates the hydrostatic pressure gradient. And from high to low, mass flow actually take place. So when mass flow take place, what happen? So water molecule move, bring together with the dissolved solutes. Okay. So we can take a photo of this and then we go to our notes. So under this model called pressure flow model, and what you need to do now, take 3 minutes to complete the paragraph here. Okay, once you have done, just in the Zoom chat, say done. Thank you. No. So the current experimental evidence supports the translocation of dissolved sugar in phloem by this model called pressure flow model. Okay, because this is due to the hydrostatic pressure, and flow means that the mass flow is not the osmosis or diffusion, very slow. Okay, the process. So first of all, we state that the flow by the means of the mass flow, and we do have the pressure gradient exists between the source and the sink. Okay, the source is where the sugar is loaded and the sink is where the sugar is removed. The sugar refer to the sucrose. Okay, so at the source, what actually happened? The dissolved sucrose is removed from the leaf of the mesophyll cells where it was synthesized into the companion cells first. So we are going to learn this one later, how. Okay, which loads this companion cell later will load the sucrose into the sieve elements of the phloem. So the loading of sucrose require ATP, so it's active transport, or more precisely, it's an indirect active transport. Okay, so the sugar therefore accumulates in the sieve element. So because of the accumulation of sieve sugar, so the increase in dissolved sugar in the sieve elements actually make the water potential lower, so decreases the water potential. So therefore, water molecule move by osmosis from nearby xylem cells in the sieve element down the water potential gradient. And because accumulation of water molecule increases the hydrostatic pressure. So at the sink, sugar is unloaded by various mechanisms. It can be active, it can be passive from the sieve element. Okay, with the loss of the sugar, because sugar lost already, so the water potential now increases. Okay, because low become more dilute. Okay, so therefore water molecule move out of the sieve elements by osmosis. Okay, and this water molecule move back to the xylem, and this water movement decreases the hydrostatic pressure because move out. So therefore, the movement, and we do know that the source and the sink can be at a different location. Therefore, the movement in the phloem is bidirectional. Okay. So if you look at this, there's something special. If you look at this diagram, you realize that water potential at the source is negative 1.1. Water potential at the sink is negative 0.4. So actually, water potential at the sink is higher than the source, but the movement of water molecule, can you see that, and solute against the water potential gradient. So it means that it's not because water potential. The movement is not caused by water potential gradient. If because of water potential gradient, it will move out, right? But this moved down. So it's due to what? Due to the hydrostatic pressure gradient. Are you clear? So if you compare negative 1.1, negative 0.8, higher, lower. So water molecule move in. So accumulate. So accumulation of water molecule, so hydrostatic pressure is high. Then you can see the water molecule is negative 0.4, higher, 0.6, lower. So move. So when water molecule move out of the sieve element, so hydrostatic pressure become lower. So move down by mass flow or bulk flow. Okay, some books use bulk flow. For us, we use the word mass flow. Okay, so when the water molecule move down, it bring together with the sucrose or assimilate. So how about in xylem? In xylem, you can see the water potential negative 0.6, negative 0.8. So higher, lower. So therefore, water molecule move. And you compare sieve plate, no sieve plate, totally broken down. Okay, now pressure flow is one of the model, In fact, actually, we do have the cytoplasmic streaming hypothesis, electroosmosis hypothesis, and peristaltic wave hypothesis to describe the translocation in the plant. Because plant, we have million types of the plant and million species of the plant. Each of them may have their own way to translocate the assimilate. But no matter which one, we do have the evidence to support and against each hypothesis. So this is not in our syllabus, just to let you guys know. What are the evidence that supports the pressure flow model? So what we have learned here. So you look at this. First, when we use the aphid, this morning we learned about this, the stylet of the aphid. When we cut it at the mouthpiece of the aphid, what will happen? The solution actually come out from the phloem, right? This is due to what? Due to the pressure already generated inside the sieve tube elements. So therefore, it shows that the tube is under pressure. Okay, and then if you sample the phloem sap at the leaf at the source compared to the sink, source will have higher concentration of the sucrose. Sap, okay, in the sink had lower concentration. So confirm that we have a different water potential, cause the osmosis of water molecule to move in out of the situ element into two locations, so generate this hydrostatic pressure. So at the top part higher because loading and unloading. So means that we know that got the loading and unloading. And if we use this radioactive tracer, for example, radioactive carbon-14, to track the movement of organic substances in the plant, then you can see that when we use the metabolic inhibitor, for example, the one that stops the respiration, when you use this, the one that stop the respiration, chemical that stop the what we call ATP productions, then you can see that translocation now stop. So it means that active transport is involved here. If no, then the process will still continue. But we do have the evidences that against the pressure flow model. If you look at the entire process of the mass flow, guys, when we talk about mass flow, is this one. This is mass flow. Okay, mass flow. So if you look at the mass flow in the sieve tube element, is a passive process. It's due to the different ingredient cannot. Okay, but if you talk about this, we still cannot answer one of the question: the presence of mitochondria in sieve tube element. Right? We do know that there are mitochondria in the sieve tube element. So how you explain this? So this model cannot explain the presence of the mitochondria in the sieve tube element. Okay, and if you measure the flow rates of the amino acids and sugar, they have different rates, and this is impossible contradict with the pressure flow model, because in the pressure flow model we know that they follow the water, so means that they must have the same speed or uniform speed. But now, why amino acid and sugar, they be transported in different what we call this speed? So means that again, this observation against the pressure flow model. Okay, so this part do not need to actually memorize it. So sometimes question may tell you this evidence and ask you which one actually support the pressure flow, which one is not, does not support. So from here, you roughly know which one support, which one does not support the pressure flow model. Okay, so we learn about this already. Now we look at what is the Phloem loading. Phloem loading is very, very important, because without Phloem loading, you won't be able to generate the hydrostatic pressure gradient. So the movement of the solute or the assimilate from the photosynthetic mesophyll cells into the Phloem sieve tube, it can be either symplastic or apoplastic pathway. So they follow water molecule. Okay, they follow water molecules. Okay, but when it comes to the companion cells, special. Whether apoplast or symplast pathway, they cannot directly enter into the companion cells. So they have to enter into the companion cells by active transport. Even symplast also cannot. Are you clear? Apoplast, they can continue, but they do nothing. They cannot enter into the sieve tube element. They cannot enter into the companion cells. So in this diagram, highlight, or say our label, this is mesophyll cell. Okay, so this is the component cells, and this is a sieve element. So what we observe here now is the cell wall of the component cells and the cytoplasm of the component cells. So this one is a cell wall of component cells. This part is a cytoplasm of component cells. Clear? Okay. So what actually happens now? I can divide them into three processes. First, second, and third. So what actually happened in the first place? So it's active loading. So means the active transport actually takes place. So ATP is used to phosphorylate the proton pump. This one, H plus ATPase is the proton pump. So ATP phosphorylate the proton pump the H plus. So the first one is the active transport. So we know the active transport we can against the concentration gradient. So therefore, you can see that we have high H plus concentrations at the wall, low H plus concentration inside the cytoplasm of the component cells. Number two, so for protons to move back, because we have high concentration of proton, low concentration of protons, so proton want to move back, they cannot pass through the phospholipid bilayer. They have to go through this, what we call the sucrose H plus co-transporter. So number two is by facilitated diffusion. So down the concentration gradient. So both of them for proton. You see that? Proton move back high to low, so down. Because using the protein, transport protein, so in this case, down the water potential gradient. You see that? Okay. Now how about sucrose? Sucrose in this case, sucrose move in also by facilitated diffusion. But it can against the concentration gradient because now H plus bring them in. Are you clear? So it's similar to something like, if you want to enter into a cinema, you need to buy a ticket. So it depends on the concentration gradient, your money. But now someone actually bring you in, so you can enter many, many times because you depend on someone. You do not depend on the money. Can you see that? So in this case, sucrose depend on the proton. As long as we have the proton, sucrose will be able to enter into the cytoplasm of the component cell continuously. So at the end, achieve this, what we call it, against the concentration gradient. Are you clear? So the whole process actually is indirect active transport. Now, why is it indirect? Because if you look at this, the use of the ATP is not directly transport sucrose. The aim here is to transport sucrose, cannot. But ATP use is not directly used. It's not directly used to transport the sucrose. We use it to transport the proton. But the net effect, proton move in, proton move back, move in, move back, move in, move back. So it means that the ATP use here is indirectly to transport the sucrose. So now we know that component cells with high concentration of sucrose, sucrose can diffuse through the plasmodesmata into the sieve elements. Okay. So what you need to do now, okay, a quick one, 1 minute to answer this question. So first, you can see that the process requires ATP. Okay, so T. So next, ATP phosphorylates the proton pumps. Okay, so another name of proton pump, H plus ATPase. Okay, so next, what happened? Next, you can see that, okay, we generate the concentration gradient. Okay, so therefore, the protons now are pumped out the component cells, proton gradient is created. Then proton can enter back to the component cells through the sucrose proton co-transporter. Okay, down the proton's concentration gradients, and finally sucrose follow. So meanwhile, sucrose molecule move into the cells through the same co-transporter against the sucrose concentration gradient. Okay, so I make a diagram to summarize this again. Yeah. Okay, so you can see it now, cell wall and the cytoplasm of the component cells. So first of all, at the component cells here, we do have the proton pump. Okay, so this is a proton pump. So what happened here is proton pump, when it phosphorylates with ATP phosphorylate the proton pump, then protons can be pumped into the cell wall. Are you clear? So I label here, this is process number, process A. So A is the active transport. And against the concentration gradient because ATP. Okay, so means the concentration of H plus is high in the cell wall, concentrations of the proton is low at the cytoplasm. So they need to go through these special channel proteins called co-transporter. So this is the sucrose proton co-transporter. Okay, so means that now protons come back. Okay, and sucrose can, okay, sucrose is green color, right? Okay, sucrose now can enter. So you label this process B and process C. So process B actually is a facilitated diffusion. Okay, down the concentration gradient, but process C also facilitated diffusion, but against concentration gradient. So means that now because of this co-transporter, so sucrose now enter. So concentration of sucrose is high, so sucrose now can move. So this one is by diffusion into the situ element. So when enter in situ elements, they lower down the water potential. Water molecule enter into situ elements, therefore increases the hydrostatic pressure. Okay, so you can take photo of this now as a summary. So sucrose now reach the sink. What they do? They diffuse through the plasmodesmata, enter into the component cells. So reverse process. Okay, so sucrose is converted into other compound by enzyme to lower the concentration and maintain the concentration gradient after they leave the component cells. Okay, so now what does this mean? So if you look at this, this is component cell. Component cells. So this is, let's say, storage cells. Let's say storage cells. So means that because of the faceted diffusions, right? Sorry, for diffusion through plasmodesmata, so now sucrose enter. Okay, at the sink. So when sucrose now enter into storage cells, we want to maintain a steep sucrose concentration gradient. They go through this transporter. So what happened here is inside these storage cells, you can see that they have the enzyme to break down the sucrose into the glucose plus fructose. Why we need to do this? First, you look at this. Sucrose concentration is high, but they will reach dynamic equilibrium because facet diffusion. So in order to do this, you can see that once enter in the storage cells, we always break down. So what is the concentration of the sucrose inside? Because we always break it down, so very low. So maintain a steep concentration gradient. Are you clear? And we break down glucose because glucose can be used in respiration. And also we can use it to, we can store it. So let's say in plants, you can have starch, okay, store it in starch. So generally, involve 3 steps: unloading. So means that from the sieve element, Okay, C2 element. So you can see that unloading take place, and then short distance travel to the storage cells, and then we store it or carry out the metabolism to remove the glucose. Okay, so by removing glucose, sucrose continue to be broken down. So therefore, we can maintain a steep concentration gradient. So sucrose always enter into the storage cells and carry out, we break down respirations and we can do the storage. Okay, so with this, I've done for the lessons for today.